right, this is the last video for week 1. This is the one on neurotransmitters and functions of the synapse. Before we get into this, just want to make sure that you review the resting membrane potential and watch the video there if it's helpful to you. The action potential, watch that video again if it is helpful to you. Make sure that in your PowerPoint deck and in the textbook, you carefully read the section on hypokalemia and hyperkalemia. That's what happens if you have too little or too much potassium. Because everything we talked about with the resting membrane potential and action potential dealt with the potassium concentrations, what happens if you have too much or too little potassium? And then, of course, review the neuronal cell structure so that as we talk about the axons and the excitatory transmission down the axon, you are familiar with the parts of the neuron. One thing to quickly talk about as we're following our action potential down the neuron. So it has formed over here at the place called the axon hillock, and then it is traveling down the axon towards the terminal end, and it's going to travel faster or slower depending on whether the axon is wrapped in a myelin sheath or not. The reason why myelination helps to move the action potential faster is because it allows the action potential to skip from one area to the next. And so myelin acts like an insulator for the ions. Since it's a voltage, when the ions will leave or diffuse away, you can cause the action potential to fizzle and be really slow. So what myelination does is it allows you to have something called saltatory conduction. That just means that the action potential doesn't have to pass through the area where the myelin is, and it just causes depolarization here, and then it can actually jump to the next node. And this space of open axon between the myelin is called the nodes of Ranvier. And at the nodes of Ranvier is where the action potential can continue, it depolarizes, and then it can move to the next area. And that just allows it to move really quickly down the length of an axon. If an axon is unmyelinated, then it moves a lot slower. So that will tell you a little bit more about the speed at which the action potential has to send the signal and what types of signals you might want to send fast versus slow. Once you have traveled with your action potential down the length of the neuron, you get to the axon terminals. The axon terminals are the ends of the axon, and they usually branch out a little bit, and they make these terminal bulbs. And at the terminal bulbs is where we might find a synapse. A synapse is the place where we're going to be sending the information from this neuron to either another neuron, other neurons, there may be more than one, or to another cell, like maybe a muscle cell, to cause a muscle contraction. So at the end of the axon terminal, we bring the action potential, and then what's going to happen is it's going to reach the end to the synapse. So let's look at that up close in the next slide. Here's a picture of a synapse. At the synapse, you have the end of an axon terminal bulb. You see it kind of elongated and a little bit wide at the end. And then you see the next cell. This is the next cell that you're coming in contact. This happens to be a muscle cell in this picture right here. So we have a motor neuron. That just means that this is a neuron that's going to be causing the action potential to spread here to our muscle cell. So our action potential is traveling down this length of the axon all the way down here, and it's going to get to this area of the axon terminal bulb. This is a voltage-gated response, right? We have voltage-gated sodium channels that are responding to the depolarization. And so we get down here. This is called the presynaptic membrane. All right, presynaptic, because the synapse is the space between this axon terminal bulb and the next cell next door. They don't physically touch. It's a true space between them. There's just some interstitial fluid in this space. And this is the presynaptic membrane, this one right here. Actually, let me put it exactly on it. There's the presynaptic membrane right there. And over here is the postsynaptic membrane. So we have presynaptic. The presynaptic has some voltage-gated calcium channels. So this is the first time that we have talked about calcium. On this presynaptic membrane, we have voltage-gated calcium channels, and they open when the depolarization reaches them. When the depolarization reaches them and those calcium channels open, they will trigger the depolarization. Let me move that forward. The depolarization opens the calcium channels. That triggers an influx of calcium to the inside of this presynaptic axon terminal bulb, so right here. When calcium influxes, so it goes in right there, these neurotransmitters, which are marked in green, in this case these are acetylcholine neurotransmitters, these neurotransmitters are going to be released by vesicles. So the vesicle will release the neurotransmitter through exocytosis to the synaptic cleft, or the synapse space here. And in that synaptic cleft, then the neurotransmitter will diffuse from the area of high concentration right here, close to where it was released, across the synapse over here to the postsynaptic membrane. So let's look at what happens at the postsynaptic. Here's a different picture, but the same thing is happening. The action potentials traveled down, and I love this one because here we see the voltage-gated calcium channel is opening in response to the action potential. Calcium rushes in, which triggers the synaptic vesicle here to release the neurotransmitter into the synaptic cleft. Very small, but a space between. Then this neurotransmitter, as it's released, will diffuse from the area of high concentration up there to the area of low concentration down here. And there's a number of different types of receptors, so this is showing you some of the different types of receptors. And each of the neurotransmitters will have its own types of receptors that will open. The ion channel then opens if the neurotransmitter binds with it, right? So neurotransmitter binds, then you might get the opening of an ion channel, or you might get a second messenger effect, which is a different type of cellular response. Either way, you may get a cellular response in this cell because of the action potential that was sent down this cell. Really quickly, before I move to the next slide, I want you to understand that the voltage change that happens over here cannot pass over here. Because they are not physically connected, you cannot have the action potential travel from one neuron to the next neuron or to the next cell that it may be nearby, because there's no physical touching between those cell membranes. So the action potential, which is an electronic signal, right, electrical, has to become a chemical signal in the form of a neurotransmitter, and then it may cause another action potential to form over here if this ion channel happens to be, say, a sodium ion channel. There's a great video that we have attached here that you can look at when you're in your slide deck. You can click on that to look at the Khan Academy neuronal synapse video. Here's another picture of the synapse, reminding you, in this case, again, we have a motor neuron. So this happens to be a muscle cell that it's going to be stimulating. So the action potential is sending it down, opening voltage-gated calcium channels, causing the neurotransmitter to undergo exocytosis. The neurotransmitter diffuses across the synapse, and then the neurotransmitter binds to its receptor. And when it binds to its receptor, it may open that receptor or may cause some other downstream responses. This happens to be a somatic motor neuron, which means this is a muscle cell, Acetylcholine is going to be this particular neurotransmitter. It binds to nicotinic receptors, causes an influx of sodium, and it causes a depolarization to occur on this muscle fiber, which is what is going to cause the muscle fiber to contract, as this depolarization causes an action potential to spread through the muscle. We'll talk about that a lot more when we get to the muscle chapter. But I want you to know that this same process happens with other types of neurotransmitters and other cells or other neurons that may be interacting with the presynaptic neuron there. What happens after you have had the signal sent from one presynaptic neuron to the next, or the effector cell? What happens then? Well, the neurotransmitters can undergo a couple processes. Some things, they may be degraded by an enzyme in the cleft. So you might have an enzyme like acetylcholinesterase. That's an enzyme that breaks down acetylcholine into component parts that are then brought back up into the presynaptic cleft, and then they can be used to make more acetylcholine, so that you just have this cycle of release the acetylcholine, break it down, bring it back in, and make new acetylcholine. So it just kind of goes through that cycle. Sometimes it may not be broken down in the cleft itself, but it may be taken right back up into the presynaptic neuron, right through that axon terminal. So the neurotransmitter, typically some of the monoamines, excuse me, might be just brought right back. Sometimes, because these are open synaptic clefts, there's nothing stopping it, a few of them may even kind of diffuse out into this interstitial space. But luckily, we don't lose too much that way. The last thing I have for you here is to reinforce that the different types of neurotransmitters will bind to specific receptors. And depending on the type of neurotransmitter will cause a specific type of receptor and the following response to the cell. So it's important that you understand, for example, that these neurotransmitters, epinephrine and norepinephrine, are called catecholamines, and they bind to adrenergic receptors. We have beta adrenergic receptors and alpha adrenergic receptors on different cells, and they cause different effects. Some other neurotransmitters, like serotonin, will actually bind to serotonergic receptors, dopamine to dopamine receptors, and acetylcholine binds to what are called cholinergic receptors. There's two classes, the nicotinic and the muscarinic, and we saw the nicotinic a minute ago when we looked at the muscle cell. So this is something to review as you finish out this chapter, the different types of neurotransmitters, whether the neurotransmitters are generally excitatory or inhibitory, meaning do they cause an action potential in their downstream target, or do they tend to make it more hyperpolarized and less likely to form an action potential, and then what types of receptors they might bind to. It's important to know about receptors and types of neurotransmitters because the vast majority of medical drugs actually act on these receptors, and they prevent the binding of different types of neurotransmitters, and therefore prevent the downstream effects on those cells by preventing the binding of a neurotransmitter to its appropriate receptor. If you have any questions about these slides or about the information, please be sure to put that into the discussion board, into the questions, or contact me by email, and we can talk about the things that you might have some confusion about. I look forward to making some videos for week 2 and to continue to work through this material with you.